I told you that from this plant, we can generate fuel to power an airplane. This is known as biofuel, an alternative to petroleum. However, the process for making biofuel is not carbon neutral, as it currently pollutes as much as fossil fuels. There is a way to improve it though, directed enzyme evolution. And the best part, we can do it in a high school lab. Enzymes are biological molecules that speed up reactions in cells, and they're coded by DNA. Most enzymes in the human body can be found in the digestive system, and they help us break down food and turn it into energy. Enzymes naturally evolve to survive in changing chemical environments through genetic mutations. Although we can now code DNA strands from scratch to generate any enzyme we want, their genetic makeup is too complex for scientists to know where to introduce mutations. Nobel Prize winner Frances Arnold found an approach to the problem, using evolution to her advantage. Evolution directs the mutations of enzymes towards increased efficiency. The better an enzyme is at its task, the more it reproduces. But how can we control this? Let's say you want to break down a molecule. The beauty of nature is that it can do this if you know how to ask. You would first identify an enzyme that can complete this task, even if it's not very good at it. This is known as catalytic promiscuity. The next step is to create a mutation in this enzyme, a process called controlled randomization. Scientists need to know which kinds of genes would be most useful to mutate, usually choosing those that code for the enzyme's active site. Then, you inject the mutated DNA into a bacteria to reproduce the enzyme and test the offspring. You choose the best performer and repeat the process until your enzyme reaches peak efficiency. So what does this have to do with biofuels? The process for second-generation biofuels, which pollute less than the previous generation, can be improved with directed enzyme evolution. Second-generation biofuels come from cellulosic material, that is, inedible parts of crops such as stems and leaves. By breaking down this material into sugar and then fermenting it, we get cellulosic ethanol, the biofuel, which could reduce greenhouse gas emissions by 60%. The pretreatment phase, where the cellulose is broken down into sugar, is the most expensive part of the process. This is where directed enzyme evolution comes in. The enzymes that are currently used to break down the cellulose are costly, and their production is ineffective. In 2019, greenhouse gas emissions from cellulosic ethanol were 97% lower than for gasoline, including those of production, which make it a viable alternative. Directed enzyme evolution is helping us find solutions to our biggest climate change challenges, and it has the potential to turn us into a sustainable society. So don't lose hope. If this generation can't solve the problem, evolution will find a way.